Take turns. We can take it out. We're all good.
Hi, everyone. Good morning, and welcome to the 28th year of the Summer Systematics Institute at the California Academy of Sciences. Yeah. I'm Dr. Lauren Esposito, and I co-direct this program along with Dr. Rebecca Johnson. And this National Science Foundation funded program is focused on undergraduate research projects. Each summer we invite mentees into the Academy from around the country to conduct original research alongside a team of mentors who are studying all sorts of things here at the Academy, from corals to frogs and really everything in between, also plants. And throughout this 10-week program, these young people have learned basically everything that you need to know to work in a museum. I mean, sure, there's probably a couple things you can learn here and there, but they've hit the ground running, and you're going to see the results of their really intensive research projects this summer. They learned all sorts of things in addition to research skills and methods, like how to communicate science effectively in a natural history museum or public museum like the Cal Academy, and how to write about science through science communication. If you missed any of their interviews on our social media accounts, you should be sure to check those out for some informal information about their projects this summer. But what you're going to hear today is their final presentations, summarizing what they've accomplished this summer, which is certainly not the end of their path here at the Academy, but just the beginning of it. Rebecca?
So I just wanted to take a moment to thank all the people that have made this summer possible. I mean, obviously our amazing interns who are just amazing scientists, as you'll hear, and researchers and communicators, but also just really lovely people and a really nice cohort of students who got along really well and just really worked as a team. And we really appreciate and love seeing a group of people that work so well together, because
Best one yet.
Best one yet. Wink, wink. But you know, that's what it takes to work at a natural history museum, right? Like, one of the things I love about working here at the Academy is working on teams and working with people who have different expertise and different life experiences. And you all demonstrated that really well as a cohort. But putting on this program, the Summer Systematics Institute, takes a lot of people here at the Academy. And so we do have a lot of people to thank. We first want to thank Ed Myers, who can give him applause. So Ed is a research scientist here at the Academy, but was hired to work with students. And this is a new way of a new model for how we run the Summer Systematics Institute and some of our other student programs. And it's been awesome, most partly because Ed is great, but it's been really nice to have someone who's dedicated and has been hired to work with you all and lead a course and help you, like, on the academic kind of intellectual side of the SSI. So thank you so much to Ed. And then I wanted to thank Arya Natarajan, who's running the live stream. Arya is a former SSI intern and works here on our digital engagement team and has been super instrumental, not only as a mentor, but also just in helping on all the communication pieces of the SSI, including today. And then I also wanted to thank Jacob Gurneau for all of his help. In addition, same as an SSI alumnus, and worked kind of in an above and beyond capacity, helping and assisting the students in addition to being a mentor. And then, of course, all of the mentors, who many of you are here in the audience, virtually or in person. Just thank you so much for taking the time to mentor a student, to share your expertise and your time and your research, and to really work with these new colleagues of yours to do research that you couldn't do otherwise. And we really appreciate all the time that you all have taken to be a part of the SSI. And then everyone else at the Academy who helped with courses and lectures and guest speaking and facilitating nightlife and just all of the different things that it takes, like everybody helping out in different little or big ways. We just really appreciate everyone's help, and the program wouldn't be as successful without everyone. I think that's it. Did I miss anyone?
No, I think that
Okay.
was good.
Okay, good.
Thank you.
All right.
The last thing I just want to leave you with is you're seeing talks from 12, 13 people this morning, throughout the day. And I just wanted to share that these are 13 folks selected from a pool of applicants that numbered over 600 this year. So really like the best of the best. And I think you'll see that reflected in their work.
All right. So without further ado, let's hear from the interns, not me and Lauren, because that's why you came, is to hear from these amazing young people. So first up, we are going to hear from Tala Al-Thaher from Arizona State University. And Tala's advisors were Laurel Allen and Arya Natarajan. And I will let Tala take it away.
Hi, everyone. Can you all hear me? Okay. Hi, everyone. Thank you all for being here this morning. My name is Tala Al-Thaher, and my project is called What's New with the Dead. And my mentors are Laurel Allen and Arya Natarajan, as Rebecca said. So there are so many science-related topics with amazing stories. So I focused on scientific collections. And let me tell you why. Before I got this internship, I wasn't too familiar with scientific collections. Even though I am studying sustainability, I've been in another research fellowship or program such as this, but we didn't really talk much about scientific collections. It wasn't very accessible to me. I didn't really know much about it. But then as I came to the academy and I started learning more about it, we started taking tours into these collection rooms. I started understanding the value to science and society. And I was like, why didn't I know about this before? And part of the reason is because it is kind of hard to access. Because obviously you don't want a bunch of random people coming into these rooms with important specimens and like messing with the jars and dropping things. But another reason probably is why people don't really talk too much about scientific collections is as I was sending these photos to like my family and being like, Mom, look how cool this is. Mom was like, what are you looking at? Like, you know, it was you get some interesting reactions from people that aren't too familiar with scientific collections or specimens and maybe don't understand exactly what they're used for. So when I was trying to set up my independent project, I had a few key questions that I was thinking about. One of the things was, how can I make scientific collections a bit more accessible for people that don't have access to internships such as the SSI or don't maybe have like natural history museums around them or that provide tours, things like that. Another way was, how can I make scientific stories engaging? So if I want to tell a story about scientific collections, how can I have people that know nothing about scientific collections think it's as cool as the people that are in it? And on top of that, combining these two things, can I make engaging scientific stories that can help shift people's perceptions on scientific collections? So these were my methods. There are a few steps I had to get to. The first step was I myself had to learn about the Academy's collection history. Like I said, I kind of entered not really knowing too much about it. And it was really interesting to be put both in the shoes of the audience that I was trying to target when I first started my project, to come out also being I'm trying to educate people. So it was cool. I got to see like both sides. So that was my first step. The second step was I really wanted to identify specimens in the Academy's catalog that were interesting, that had something, that had heart, that would get people interested and engaged, and that applied to a conservation arc, because I think that's what really gets people nowadays, and that was the lens I wanted to come in with, or the angle of scientific collections are great for conservation. How do I show them that? What specimens really show that? My third step was to create a story map that brought it all together, and I'll touch on story maps in a second. And my last step was I wanted to test it with a target demographic of 18 to 25-year-olds. So the reason why I chose 18 to 25-year-olds is I feel like they are the next generation of potential scientists and decision makers. And if I kind of get it in their head as early as I can, like, hey, scientific collections are important, and this is why, and this is how, then maybe like when they do end up in those roles, they can be thinking about it or kind of have it in the back of their minds. So why story maps? So story maps are interactive. So this is just a snippet from my story map. They're digital, they're portable, and they're mobile friendly. So this is on a desktop, but you can also look at it on your phone. Story maps are multimedia platforms that allow you to have like visuals in them, maps. So here on the screen, you can see that I have like a map of the Galapagos, and on those little location markers, I put like fun facts for people. So you can choose to engage with it, you can choose not to. And they help accommodate lyrical writing that I think is more immersive than just articles and essays. Because if I give people like a chunk of text and be like, this is why scientific collections are important, sure, if they read it, then maybe they'll get something out of it. But the audience I'm trying to target is probably won't be reading it or won't stay as engaged. Maybe they'll read like the first 3 lines. But with story maps, I can kind of have like a photo with text, and breaking up that chunk is maybe more digestible for people. So what's new with the dead? This is a little sneak peek into my story map. This is the, the opening page. So what's new with the dead? The centuries-long effort to protect nature. I really like the title because I feel like new and dead kind of contrast pretty well. Like it kind of gets people thinking like, oh, what is new with the dead? What dead are we talking about? Like, am I talking about vampires? Am I talking about like what exactly am I talking about? You know? So I'm gonna walk you through some parts of my story map. So we open onto the 1905 Academy expedition to the Galapagos. And this story was really important to me. So these are pictures from the Academy's archives. So this is actually photos from the 1905 to 1906 expedition to the Galapagos. So this is Rolleback and his crew. In the photo on the right, you can see they're transporting a tortoise they're trying to get it to their little like boat so that they can take it to the ship because they were really heavy. The photo on your left, your bottom left, is they are collecting, I think they were collecting birds. And then on the top left, they are, those are marine iguanas. Yeah. So I wanted to open up with this story for many reasons. The first reason is I wanted to highlight kind of how collection used to be. Like it used to be like, or the ways that it was extractive in the past. Like so this expedition, they came back with 76,000 specimens. And I think for somebody that doesn't know about collections and they see this, they're like, oh my gosh, that's so many. Like, why are you taking so many? And it's true. Like those are valid concerns. And I didn't want to shy away from any concerns or criticisms that people have had. I actually wanted to be like, here's the history. Like this is true. It's happened. And I wanted to show how to segue into how whatever they collected is still being used today. So that leads me into talking about the vermilion flycatcher. So the vermilion flycatcher is a really interesting story that Dr. Jack Dumbacher and Dr. Mo Flannery helped me find out about. And so the picture in the middle is a photo from the collections from 1905 to 1906 Galapagos expedition. And these birds recently, a team at the Academy did a genetic analysis of these birds, and they found out that there were actually 2 species in the Galapagos, and that one of them is most probably extinct in one of the islands. So this was a really cool story because I didn't know anything about vermilion flycatchers. It really tied in nicely to the Galapagos expedition and how I'm trying to highlight the ways that these collections are still used no matter how old they are, like they're over 100 years old, but look like they're helping us discover new things. And how studying the genetic makeup of these, why is it still valuable today? It's still valuable today because understanding the genetics of this species can help us relocate the closest genetic relative to the islands and help bring back that biodiversity. So that was really important to me for people to understand. And not only that, but the Academy scientists don't just collect specimens from places around the world, but they also collect close to home. And this is where I talk about the Xerces blue butterfly. And so I'm not local to San Francisco. I'm not even local to California. But when I was talking, I interviewed Chris Grinter for this story, and he told me that San Francisco was sand dunes. And I was like, what? Like, I had no idea that was a thing, because I think when people that don't know about San Francisco come into San Francisco, they just think about the Golden Gate Bridge. They think about like rolling, like the hills of Victorian homes. They never really think about sand dunes. So this was also a really interesting story. And the opening line to this section is written over there on the top left. Once upon a time, San Francisco was a desert with rolling golden sand dunes. No, really. And I kind of wanted to come into this being like, I'm also discovering this with you to the readers, but also help them understand about Xerces blue. So I got lucky that there was a story with a butterfly, because butterflies are charismatic, and I think anybody in the public like really loves learning about butterflies. And the Xerces blue is a really interesting story because it is extinct, and it was endemic to San Francisco, but the Academy does have 500 specimens in their collection, and what's really cool is that they can still use these. Again, it's a similar story to the vermilion flycatcher, where they're trying to understand the genetics to see what's the closest relative to bring back to San Francisco to help with that biodiversity, because an ecosystem that has more biodiversity is more resilient, and that's what we want. And then finally, historic collections are instrumental to contributing to our understanding of nature today, and they can directly help protect our species under threat. And this is where I talk about the coho salmon. So this story was also really cool, and Dave Catania was really instrumental in this story. So the coho salmon, their story is a little funny because back in the early 2000s, this timber company wanted to delist the coho salmon from threatened to not threatened, because their argument was that, pre the early 1900s, these coho salmon didn't exist in streams south to San Francisco. And they were like that they were a product of, like, I forget the word, but hatcheries in the area. So that was their argument. But then the Academy actually has found 17 specimens dating to the late 1800s of these coho salmon, and that totally changed the game. And so this went to court. It went to the U.S. Fish and Wildlife, and instead of delisting the coho salmon from threatened, they actually bumped it up to endangered. So that completely restricted even more what the timber company and other timber companies could do there. Obviously, they were not very happy, but we don't really care about what they think. We're just happy that the coho salmon can be safe, and that these streams are now protected, because the coho salmon is not only important to freshwater, but it's also important because they go out into the ocean at some point. So they're very good sources of food. Their bodies, because coho salmon go back into freshwater, the females, after they spawn, and they die there, which is a great source of nutrients. So that's a really great story that I think helps highlight how it informs policy and how it continues to protect our species today. So my takeaways and my next steps. So there, me on the left there, and also on the right, I have a few people who were helping me test my my survey. I created a survey to understand, like, again, the perceptions before and perceptions after people read the story map. And the QR code in the middle is a link to my story map. So I sent out my survey to RU students. A bunch of directors were very kind enough to help me distribute it. But I also tested on nightlife, one of the nightlife events that we have here at the Academy. And I had a button making machine, which I really think was the driving force to people coming to my table. They wanted to make a button, but I had a ton of people that knew nothing about collections really come and talk to me. And based on anecdotal evidence from what I've spoken to them, I'm pretty optimistic that people left the story map knowing more. I got a ton of feedback talking about how they really liked the way the story map was set up, like how it was interactive, and also how they learned, like, again, San Francisco had sand dunes. And even if their perceptions didn't shift like hugely, if they're not necessarily thinking about scientific collections, I know that they came out learning a lot more. And I think that that's still very valuable and important for me to know. And some challenges I had was the vermilion flycatcher story was pretty hard to write. Just for when I was doing these interviews, it's a lot of scientific jargon, and like I understand it. And I'm sure all of us understand it here in this room. But I was like, how can I get people that don't understand? Or maybe, like, if I include a species name, is that going to take them out of the story? Is that going to keep them in the story? How do I keep them, like, wanting to follow along? And so that was really interesting. I had to keep thinking about my audience, and that was very valuable and very interesting. And I'm very happy that I created the story map, and I hope to continue to test it and do the data analysis as the weeks come along, and create maybe another story map like this, because I think they were super fun to make. The story map platform is cool. I wish it was a little more, it's not as fluid as I thought it would be, but it's still pretty cool. You get maps, you get pictures and texts. So yeah, not too bad. So thank you all for listening. I just wanna, so the QR code again on the top is to my story map. The one on the bottom is to my LinkedIn. I don't know, I think people think LinkedIn's important. I wanna thank the California Academy of Sciences, the SSI program, for this amazing internship these past 2 months, and the NSF, and to my own program, UC Santa Cruz Doris Duke Foundation, Doris Duke Conservation Program, for helping me find this internship, as well as my mentors, Laurel Allen, Arya Natarajan, the amazing 3 people we have up here, Dr. Lauren Esposito, Dr. Rebecca Johnson, Dr. Ed Myers, to everyone that I got to interview, Dr. Jack Dumbacher, Chris Gunter, Dave Catania, the nightlife team, Lynn Kung and Richie Lipton, as well as all the people that were in B2 basement. It was rough down there. Amy, Joy, and Luigi. You guys did not want to come down there the last 2 weeks. All the collection managers, you really helped me. Love collections so much. I have to find another natural museum in Arizona just to stay involved. And to my SSI 2023 cohort, you guys are amazing. So thank you all for listening. Now I'm gonna hubble down. Oh, I forgot questions or a thing. Okay.
If you have a question, I'll come around with the mic so people online can hear you.
What would you say is the biggest thing you learned here in your time?
That's a great question. Honestly, I'm gonna say that based on the internship that we were in and our cohort, I learned a lot about community, which is really nice. Not only just the community that we have as a SSI cohort, but also the community within the academy itself. A lot of this work is collaboration, and we always talk about that, but I think seeing that in real life and seeing how that plays out was really interesting and really valuable, and I'm taking away a lot of like cool skills, social skills to back home, and hopefully I'll be able to help build another community in other places.
We have a question from one of our online viewers. They want to know, what was your favorite part of making your story map?
My favorite part of making the story map, I got to be really creative with it, which was fun. I got to like look up a bunch of pictures. My favorite thing I think was making the maps. I got to make like a bunch of Galapagos maps, which was fun. And the story map helps you make them, but it was cool to be like, okay, where exactly am I trying to set people? Where am I trying to orient them? And I think that was really important to the narrative as well. So I got to think of it from a creative way as well as like a storytelling way.
Were there any other communication platforms you were considering besides story maps?
Honestly, no. I'm familiar with story maps, so that was kind of my go-to. I think that it was once I knew it, I think it was the best decision, or the one that I knew about, because I already have some skill with it, so it wasn't too hard to learn or like get familiar with again. And I think it's really cool. It's a really cool platform, and not many people know about it, which I think is also part of the appeal. When I had people coming up to me, as I was like, hey, do you know what a story map is? And they were always like, no. And I was like, okay, great, now you can find out what it is. So it was like it was helping people learn about 2 things simultaneously, which was cool.
Awesome.
Okay.
I just want to say super quickly, let's give an extra round of applause to Tala, who delivered this with a fractured foot.
Thank you.
All right, thanks so much, Tala. Next up, we have Amy Wu from the University of California, Berkeley. Amy's also a science communication intern, and her advisors were Laurel Allen and Arya Natarajan. Hold on one second. It's just the green arrow, right? No. Okay. One moment, please.
I can't see it from over here.
Do you want me to make it full view? Woohoo. All right. Thanks, Lauren. All right, here you go, Amy. Thanks.
Hi, everyone. Thank you, Rebecca, for that wonderful introduction. My name is Amy, and today I'll be talking about my summer research project, which was essentially creating a mini science comic series entitled The Field Scientist. So within science communication, we are really interested in how do you get the general public engaged and excited about science. And in particular, we really want to look at communities and groups of people who have historically been marginalized or excluded from science. How do you get them to feel like there's a space for them in STEM, and then to get them to actually thrive in STEM? And so according to the National Science Foundation, people of color, women, and LGBTQIA+ identifying folks continue to be underrepresented in STEM. And when they looked at primary and secondary education, it was found that high school students who actually reported feeling a greater or more positive science identity and sense of ability were more likely to go on to actually major in a STEM undergrad degree later on. And so this project really endeavored to look at to identify novel ways to foster a sense of belonging and personal interest in STEM among these secondary school students. So we decided to focus on science comics. So science comics have become increasingly popular in the most recent decades. You can find them published from places such as the World Health Organization, NASA, and journals like Nature. They are traditionally basically take a sort of complex scientific phenomena, distill it down through illustrations and narration to make the science more accessible for a broad public audience. And so my comic series, The Field Scientist, implements those sort of traditional characteristics of science comics, but then also intertwines biographical narratives so that we can explore how the lived experience of scientists helps inform the types of science that they decide to pursue. So these were sort of our key research questions. And just as a whole, our goal was to essentially look at how we could center the experiences of scientists who have intersectional identities and how we can explore how their lived experiences as scientists at different points in their career help impact the types of science they do. And hopefully in sort of seeing this journey, people, especially secondary school students who are reading these comics, can envision themselves in science. So as I sort of had briefly mentioned, I sort of envisioned this mini comic science comic series entitled The Field Scientist. And so every single comic follows the sort of biographical narratives of one scientist that I got to interview. And so we did one primary interview that was about 1 to 2 hours, and then we had secondary correspondence via email or additional interviews. And we worked really collaboratively to narrow down the narrative that we wanted to share and the themes that we felt were really prevalent within their lives. And then also worked collaboratively to figure out the illustrations that we wanted to create. And just on sort of a side note, when I first thought of this project, I was really interested in field science because I think field stories are super exciting and there's like a lot of adventure, like there's a lot of twists and turns. And it seemed like a really good story to tell within the comic format because, as I'm sure most of us are aware, comics, especially in the US, typically tell superhero stories. And I think scientists are in their own ways sort of like superheroes. But as we started to interview, what we found was that it was actually the discussion about identity and belonging and inclusion and diversity in science that felt significantly more substantive and impactful to our readers. And so the sort of focus shifted from let's tell these really fun stories about science, field science and get students excited about field science to let's really focus on lived experiences and the stories of the people actually doing the science. And sort of in that journey, we found that field science stories actually are a really great vehicle to talk about these like issues about identity, because these are moments in which scientists are forced physically to leave their sort of ivory towers, whether it's academia, a natural history museum, or another research institution. And while historically science has been really good at pretending that science isn't informed by social, cultural, political, historical systems and structures, that's really hard to ignore when you're out in sort of the quote unquote real world and actually interacting with other people. Oops, come back. There you go. Actually interacting with other people and with other ecosystems. And on that topic, we found that in sort of interviewing the scientists that we selected, comics also are a really great place to talk about really complex and difficult topics. So these scientists experienced things such as violence against Indigenous communities. Oh no. Violence against Indigenous communities, systemic racism, considering LGBTQIA+ safety in the field. And these can be really complex and sensitive topics to discuss, especially with secondary school students. And so by sort of combining lyrical narrative as well as creative illustrations, we're able to talk about these in a respectful and engaging manner. So Good Fire was the very first comic that I created for this series, and it follows the coming-of-age story of Nicolaus Cruz as he sort of goes from being afraid of fire as a child to doing his undergrad, and now as a PhD student, working collaboratively with an intertribal nonprofit as well as a local indigenous tribe to revitalize cultural burning in Clear Lake, California. So this is Nicolaus Cruz. Nicolaus is a fire ecology PhD student at UC Santa Cruz, and he works specifically with the intertribal nonprofit TERRA, which stands for Tribal Eco Restoration Alliance, as well as the Robinson Rancheria tribe at Clear Lake to understand the timing and intensity of cultural burning. And so some of the themes that we really got from our interview with Nicolaus was that one, science can combine a lot of different disciplines. So he comes from a social science background, so we were showing that field fire ecology itself is sort of potentially a new type of science for these students, but also that science doesn't have to be just pure science. We also felt that for him, science was incredibly personally meaningful. So his personal background, his personal connections, I promise I'm not clicking the black thing all the time, but it just keeps disappearing, informed the types of science that he wanted to do. And also in his collaboration with TERRA and the Robinson Rancheria tribe, he was holding multiple ways of knowing. So both being trained as a Western scientist, but then also engaging with indigenous ways of knowing and interacting with the natural world. And then lastly, his experience of feeling a sense of exclusion because he was asking these socially informed scientific questions that his peers in his undergrad institution weren't asking. In the process of creating this story, we got the consent and encouragement to actually share this broader, important historical and cultural story about tule, which is the wetland plant that Nicolaus studies. And so this is the history of the Bloody Island Massacre, and this was an opportunity to tell an indigenous story not in the framework and lens of trauma, which I think is typically how these stories are told, but to rather really focus on the agency, survival, and resilience of these people, and in particular Lucy, who's pictured on the screen, who survived this massacre by hiding underwater and breathing through a tule stem. And her descendants are now part of the Robinson Rancheria tribe. And so through this additional story, we're able to get at how science is very culturally and socially important, as well as historically informed, and that science can be incredibly collaborative. So scientists and local communities can create these really mutualistically benefiting relationships that make science not extractive, which is how it's historically been practiced. So after creating the comic, we created a pre-comic and post-comic survey, which included a set of core questions. We tested this via snowball sampling, so both digital and in person. And some of the limitations of this sample size are that it's pretty small, especially when we start to parse down to the actual target demographic that we want to look at. And additionally, it was biased. So CAS people are wonderful. CAS people also very much like science. And similarly, the summer programs that would let me share my project were also tended to be sort of more STEM oriented. So hopefully with more broader sampling, we can get a sample size that is more representative of the general public. Okay, so who was our audience? So we found that actually a third of our respondents fit within that 10 to 18 age demographic that we were really interested in. And when we looked really briefly at the demographics of gender identity and race and ethnic identity, it was pretty diverse overall, and that stayed relatively consistent as we sort of started to parse down and look at more narrow subsets of our demographic. But we are particularly interested in 10 to 18-year-olds who we identified as sort of currently non-STEM oriented. And the way that we did that is we took the average value of their answers to 2 demographic questions, which is shown on the x-axis. So they were asked to respond on a scale of 1 to 5, 1 being strongly disagree and 5 being strongly agree to the statements, "I love science and I want to pursue a science career." So if their average values were below 3, then we included them in this sort of non-STEM category. And that resulted in around 19 survey respondents. And they sort of form the basis of most of my data analysis, with the exception of this first question. So first we want to look at, are comics engaging? Like, do students within this age group even like comics? And so we asked both pre and post comic a survey question asking them to rank their most to least favorite way to learn science. And so as you can see, this is the pre-comic results. So relatively evenly split across the spectrum from a ranking of 1 to 5. Comics are at the very bottom bar. But if you look at just the 1 to 2 rankings, so these are students who said comics would be my most favorite to my second most favorite way to learn science. Comics were actually ranked second, right after websites and tied with magazine articles. But also, if you look at the rankings for number 5, which is them saying this is my least favorite way to potentially learn science, they also had the biggest group there. After reading the comic, so there were sort of shifts all across the board, but surprisingly, we found that for comics in particular, the number of people who ranked comics as 3rd, 4th, and 5th actually increased, which was not so great for us. But we decided, okay, let's actually deep dive into the data. And what we found is that we decided to compare these survey questions to a demographic question where we asked, how often do you read comics? And it was pretty interesting because we found that students who said they never read comics actually picked comics as their favorite way to learn science after reading it. And so this suggests that for these students, comics just in general are a really novel and exciting way to learn information. On the flip side, students who said they consistently read comics actually ranked comics as their least favorite way to learn science. And so it might be that for students who love to read comics, they just want to escape and not think about school or education. And so adding this educational angle within a medium that they really enjoy actually doesn't make it that much more engaging. We also wanted to sort of gauge perceptions of science both before and after reading the comic. So specifically looking at our target demographic, these are word clouds. So the bigger the word is, the more common that came up as a response. So you can see that after reading the comic, nature, learning, hard, and smart popped up more. So nature and green likely are in reference to the ecological work that Nicolaus does, while learning, hard, and smart we felt was attributed to how he shared his experience and his personal academic path. And we also noticed that words like lab, speaker, research, and experiment popped up significantly less or completely disappeared after reading the comic. So it seems that overall, the comic was changing general perceptions of what science is like and moving it away from a traditional lab science sort of image. When we asked this specific question, and these were the answer choices that students could select from, we found that overall there was a greater shift towards recognizing science as personally informed. So the absolutely and the yes answers increased post-comic, which is the bottom bar. But the maybes and the definitely no's also decreased, but the no's actually increased. And so when we did a one-to-one comparison from the pre-comic to the post-comic survey, we found that people either kept the same answer or they completely flip-flopped to the opposite end of the spectrum. So some possible explanations for this include that for the no's, we didn't specify what that way of learning was in our explanation. So it might be that they thought everybody should learn the way that Nicolaus learns and being very interdisciplinary. And it could also be that they just didn't read that sort of explainer. And so when they clicked no, what they really meant was it shouldn't matter who you are. Everybody should be able to be science and be treated respectfully and equally. Our last quantitative question of the day, could you imagine yourself as a scientist? So super exciting. When we look at the pre versus post comic surveys, the number of yeses actually doubled. So we went from 7.7% to 15.4%. And so it seems that there was a broad shift towards people sort of inching their way in this yes, absolutely direction. And with regards to this like slightly bigger no section, which seems to be the theme of this data, what we found is that when we did one-to-one comparisons, people were either keeping their answers the same or they were moving like one category closer to the yes area. And so this no section likely represents people who either didn't change their answer before and after they stayed no's, or they were originally the definitely not area and are now sort of bunny hopping their way to the no's. So small progress, but I'll take it. Okay, so we also asked this open-ended question, how do you feel after reading this comic? Starting off with our target demographic, we found that 12 of our 19 respondents actually shared sentiments of feeling surprised or impacted or like they learned something. So this is really exciting because it's showing that to a certain degree, like this comic was engaging and it was sparking a sense of curiosity in them. Looking at students still within this age range, but that are inherently interested in science, we can see that they're sharing very similar sentiments, but they're also expanding and starting to get at some of sort of the core themes that we had within Nicolaus's story. And then lastly, looking at just sort of everybody, we can see that again, people are reacting pretty positively and are really starting to sort of tease out these themes of science being very socially, culturally, politically, et cetera, informed. And so overall, it seems that respondents within all age groups and within STEM versus non-STEM, were able to reflect and actually get at some, if not all of the major themes that we wanted them to. So our major takeaways, quantitatively, it seems unclear whether or not science comics are actually effective, but qualitatively, the open-ended responses seem to show that they enjoyed it. Highlighting less visible forms of science helps respondents reconsider what their initial perception of science is, and that centering the stories of diverse scientists directly helps students within our target demographic feel a better sense of belonging and accessibility within STEM. And personally, this project was very much informed by my experience with science. I loved it as a kid, got really scared at Berkeley and like the academic culture there, and then refound it later on when I was a senior, and it was actually through a field science class. And that's because for me, field science really helped spark a sense of curiosity and childlike wonder. And it seems that generally that is this major sentiment that sort of respondents walked away from. And that's how I think you get people to love science, is to sort of really get at that innate sense of wonder and curiosity. And so that's really exciting. Lastly, looking forward, hoping to make more comics. So sort of, if you want to be interviewed, let me know. And also looking to broaden and improve survey testing. So hopefully getting a better sample size. And lastly, but certainly not least, thank you to everybody on this slide. I don't have time to thank everyone personally, but y'all have really made this project possible, 1, and 2, this has been like the best summer of my life. So thank you so much to everybody on this screen. Yeah. Yay.
Hello. First of all, that was amazing. Good job. My question is, how did you create all of the graphics for the comics?
Yeah. So funny story. I actually like took a hiatus for about a decade from drawing. So this was also me like relearning how to draw, which was really hard. But I used Procreate on an iPad. And it was just a lot of like, this is a very scrappy done comic. Like it was a lot of trial and error and like Googling stuff. And I think there are a lot of things about that platform that I want to learn because it would have gone a lot faster had I known sort of those tips and tricks. But it was a lot of that. And also like shout out to Sadie and Arya who are in this corner, who helped me a lot with like getting or trying to like troubleshoot Procreate and figure out what exactly the compositions I wanted to create were. Thanks.
Thank you for this talk. Where can we find your comic now?
Oh, yeah. Hold on. Briefly. There is a bit.ly link. I am waiting on consent to actually publish this because this is not my story. Like I'm not comfortable sharing it on like a public platform, but another future goal is to hopefully actually like post these comics on like issue.com or like a social media account or website. But for now, this bit.ly link is where it lives.
Any questions online?
first of all, you did amazing. And for y'all who haven't read the comic, y'all have to do it. It's really engaging and impactful. Anything else, like any last comments that you weren't able to talk about this, but was really impactful just because of time?
Yeah, I mean, I think there are a lot of aspects of this project that I found very personally meaningful, like both in rediscovering science and comic and arts. I think these are all things that as a kid I really enjoyed. And then like life gets busy and there are like social pressures to like do something else with your life. And I think for me, like this internship has helped me grow and reflect a lot. And a part of that is recognizing that as an adult, like you have the support and like the confidence and the space to like give your 5-year-old self permission to do all of the things that she really loved. And that at that time, like you couldn't support her in that way. And so I think it's just been really personally meaningful, and also toggling off of what Tala said earlier, like y'all are amazing. And it's been so great getting to know everyone and just seeing everybody like blossom as a scientist. Doing all the live stream interviews has been like one of my favorite parts of the summer. So y'all are all really great. Cool.
All right. Thanks so much, Amy. I have to like get the little tears out of my eyes after that little. All right. Next up, we have Joseph Wu, who comes to us from the University of California, Riverside, and his advisor is Dr. Rayna Bell. All right.
Hello, everyone. My name is Joseph Wu, and this summer I was a scientific illustration intern working with Dr. Rayna Bell on a variety of projects, which I'll be sharing with you today. So what is scientific illustration? Scientific illustration covers a wide variety of different contexts, including scientific publications, public communication and education type material, and branding and identity for labs and organizations. So as you can see from my itinerary, my project sort of spanned the range of all of these different contexts, starting with the illustration I did for species description for a newly described species of Hyperoleus. So what is Hyperoleus? Hyperoleus is a genus of reed frogs in the family Hyperolidae, which are found in sub-Saharan Africa, as you can see on this distribution map. On the left is a picture I took from the Public Floor in the Colors of Life exhibit, showing some of the challenges in identifying and describing new species in this genus. As you can see from the big leaf in the middle, there are 3 different color morphs within this one species, which can make it very hard to tell what's what, and that can be dependent on the stage of life, the sex, or the geographic location. So our specimen was found here in the country of Equatorial Guinea, which is outlined in red. As you can see, Equatorial Guinea lies in a dark green area, which indicates high diversity for this family. But most of the sampling for this genus comes from the neighboring countries of Cameroon and Gabon, which are to the north and south and have similar geographies. But understanding the particular distribution of Equatorial Guinea is important because of economic change in the country happening due to the discovery of oil offshore in the 90s. So the country has been undersampled due to a number of historical and political barriers. Our sample is on the top left. And as you can see below it, this is another sample of Hyperoleus found in the same region in Monte Allen National Park, which is in the inland mountainous region of Equatorial Guinea. None of them really look very similar to our specimen, so you can tell right away it's probably something different. It also has a different body size, so ours is around 35 millimeters, where other males in these species of Hyperoleus sampled are about 20 to 30 millimeters. So right away you can tell it's something different, but then we reach the challenges of actually describing it as a new species. So our herpetology curators, Rayna and Lauren Scheinberg, only had 48 hours in this location, and they were unable to get great photos of it in life. They were also only able to find one representative sample for the species, which is a male, which also means we don't really know what the female looks like because there are sexual dimorphism in this genus. And another challenge in describing species is identifying relatives for close relatives for comparison. So when you describe a species, you ideally want to be able to compare it to its close relatives to figure out what morphological traits distinguish it from those relatives. On the right is a phylogeny Rayna generated using the 16S mitochondrial DNA barcode and nuclear sequences. And our species is this little red line at the bottom. As you can see, it's clearly nested within Hyperoleus and not any of the other genuses within Hyperolidae. However, if you look a little closer at Hyperoleus, you can also see that it's not particularly closely related to anything, which means we don't know what we need to compare it to. And if we were to do a comparison, we would have to look at dozens of other similar species. So the next step for this project would be to do a nuclear gene sequence to better resolve the evolutionary relationships for our new species. As to my part of this project, I looked at other examples of species illustrations from field guides and books. Yeah. And what's typically drawn for frogs is the full body dorsal view, a closer view of the vocal sac, which is used for calling in males, and not pictured here, but a close-up of the hands and feet. So I took close-up photos through a microscope of all of these features, and then I sort of stitched them together to get a full body picture, comparing it to a better full body picture to make sure all the proportions were correct. And then here's my final illustration. So some features we paid particular attention to are the nostrils, which are used sort of as landmarks for cranial measurements. The curvature of the snout, which is slightly concave in comparison to other species of tree frogs found in this area. The tubercles and toe pads on the hands and feet, and the vocal sac, which sometimes contains a gland, but as you can see in ours, it's pretty smooth. So working on this project gave me a better appreciation for the diversity of frogs sort of within and between different species and groups. As for my second project this summer, I contributed diagrams and figures for an ebook on visual biology, which Rayna is working on as a larger part of an NSF grant on frog vision. So this ebook is interactive and contains clickable elements. As you can see on the bottom right, you can click on these captions and they'll pop up different labels and descriptions. So we wanted to create diagrams that would be well suited for our target audience, which is high school and college age students, while also having these interactive, fun sort of elements to help keep people engaged. So here's an example of a diagram I contributed to this project. And the top is our reference, and on the bottom is my version. So a few changes we made were more clearly showing the difference between clear and murky water. So this is showing light absorption and penetration in water. Light tends to penetrate better in clear water, and blue light tends to penetrate further than red and purple light, which is more absorbed. However, in muddy water, red light penetrates further than any other color. So this difference is more clearly shown in my version of the diagram, and you can immediately see the clear versus muddy difference. And we also have a fun little frog in the corner. So here's another example of a figure I contributed to this project. Thank you. On the top is a figure taken from a scientific journal article about the effects of different light environments in urban versus natural light. So on the in the original figure, it's all combined, but the blue line shows the curve for moonlight emission spectrum, and the brownish green orange shows artificial light. So I split this into two. So on the left we have natural light, and on the right we have artificial light. Here you can see that artificial light has a higher irradiance, which means it's brighter, and it also has a different selection of wavelengths. So orange and yellow light are relatively larger in proportion, where moonlight is more well spread out across the spectrum. And you can also see how it sort of affects frog behavior. So our frog on the left looks a lot happier than our frog on the right. So this is just a select handful of the different figures I worked on for this ebook, the more fun ones. So what the particular considerations we took into this project were mostly the target audience. So how do we keep the audience engaged? How do we deliver the appropriate amount of information for our target audience, which is high school and college age students? And then just making them fun, fun to look at. It was definitely a different kind of project than my first one, which is more descriptive and literal. And this one, there's a little more interpretation and simplification. So my final project for this summer was to redesign the CCG logo, which is the Center for Comparative Genomics here at the Cal Academy. I was commissioned by Dr. Athena Lamb, who's the director of the CCG. So here's where we started. The original logo for the CCG, as you can see on the left, was just a double helix with some text next to it, which I think is perfectly fine. But we wanted to better represent the kind of work we're doing here at the Academy, which is centered around biodiversity. So I had a meeting with Athena and took a whole bunch of notes about what we wanted for the logo, including representing at least one species for each of the departments that work with the CCG, and really representing the biodiversity and genomics, like both sides of that. Another consideration was also how we were going to use the logo. So we knew we wanted to use it on a website, and we also knew we might want to use it for merchandising, like shirts and stickers and stuff like that. So that means it needed to look good both in a larger form and in a smaller form, shrunken down, which was a challenge because we wanted to also fit a bunch of different species into the logo. So we were a little bit worried about how that would look and if it would translate very well. So I started collecting references. So I ended up with a whole folder full of photo references on my laptop. And I also ended up with a few physical specimens at my desk. So on the left is a little box of weevils that I still need to return. And then in this little sauce cup is a live scorpion that was handed to me in the arachnology department, which I think is mine now. I think I gotta adopt it and take it home with me. Yeah, so I ended up with a whole collection of little things at my desk. And I also went and did a Google search of what's out there. So a lot of the logos out there sort of have the similar theme of just including a bunch of different species and then throwing a tree in there for like the tree of life motif. So that's where I started off with my first draft. I have a cycad at the top, which is the tree looking thing. Is it a tree? I don't know. Which is the big plant on the top. And then I have a bunch of different species scattered across the bottom, representing the different kinds of work we do here. And then I just threw the double helix, which we knew we wanted to incorporate across the middle. But I felt like that was a little bit generic. We've seen stuff like this before in the Google search. So I came up with the second draft, which better incorporates the double helix into the design, going diagonally across. And then we have the different species sort of in the negative space to carve it out. So here's where we're at now. This is not a final version because I still need to polish it up a little bit. But I think we ended up with something that represents a lot of the different long-term and ongoing projects at the CCG. And it also does look good when shrunken down into a logo size like the original logo. And hopefully it'll represent the sort of things you want to show at the Academy and like the identity and branding of the CCG. And finally, here's my acknowledgements. Thank you to the Cal Academy for hosting us, and to the Maxwell Hanrahan Foundation and Robert T. Wallace Endowment for funding my position. Thank you for the NSF for funding everyone else. Thank you to Dr. Rayna Bell for being an amazing mentor. Thank you to Lauren Steinberg and Ed Myers for welcoming me into the herpetology department here at the Academy. Thank you to Rebecca Johnson and Lauren Esposito for being our program directors. Thank you to Rachel Diaz-Baston for advising me on scientific illustration careers. And thank you to all my peers in SSI 2023 cohort for being amazing. Any questions?
So what was the hardest project for you, and which one did you enjoy the most? I guess it's two.
I was hoping nobody would ask that because I have to pick favorites. The hardest project was definitely the CCG logo, just because I had a lot of creative direction on that project. And obviously there was a lot of different species that I wasn't particularly familiar with being included in the logo. But it was also very fun to work on just because I got to hear about like the wide range of different research going on at the academy, which is like really cool. And people are really enthusiastic when talking about their species. So at some point you were there, but I got mobbed by the nudibranch people at my desk. Yeah, they like shoved a book in front of me and asked me, "What's your favorite one? Which one do you want to put?" Yeah, so it was very fun to work on, and I had to, but it was a little hard because I had a lot of creative direction and freedom, which they gave me a lot of trust on that project. I really liked working on all of them. They were all sort of scratched different itches, so that's my political answer. I guess.
So are you hoping to keep going in scientific illustration? Like, is this now your potential future direction?
So I have sort of like a hard science background, and I'm doing a master's starting next fall in biomechanics and gecko biomechanics. So I was thinking, well, I'm more of a research person, and I really want to do a research career, but I definitely think like scientific illustration will be a big part of that, like in the future. So.
I have a question from the chat. Kate Montana wants to know, do you prefer drawing live animals, dead specimens, or from a reference photo?
Oh, I end up relying on reference photos a lot. Thank you to all the photographers out there. Live specimens are a little hard to draw from just because they move usually. And sometimes like the thing with museum specimens is sometimes they don't really have the right color or body posing, but it doesn't really look lifelike, I guess. So yeah, I guess my answer is photos. Oh, this is especially a problem with herps because they tend to just be brown in collections and have this kind of body pose. So.
How did you at last settle on the arachnid you picked? I noticed it was a pseudoscorpion, and I know that that was a matter of some debate.
Okay, yeah. Proctor and Harper in the back, they could answer this. They were the ones who suggested it in the end. So if you have any complaints about it, you can go take it up with them. I don't know. I took their word on it. So I deflect responsibility.
All right, we're gonna take a brief bio break and resume at 11:20. Let's give Joseph another round of applause.
All right. Hi, everyone. Welcome back. We have 3 more talks before our lunch break. So let's get started. Our next talk is Arlene Vargas, who comes to us from the California Polytechnic State University at Humboldt. And Arlene's advisor is Dr. Rayna Bell. Come on up.
Hello. Hi, everyone. I'm Arlene, and thank you for the introduction. So this summer, I was part of the herpetology department, and my mentor was Dr. Rayna Bell, and my project is titled, Who Is That? Using Genetic and Morphological Data to Identify Cryptic African Tree Frog Specimens in Natural History Collections. This is the tree frog. So what's the importance of natural history collections? They are resources for research and conservation. They are historical records that provide a biodiversity baseline that allows researchers to track and record temporal and geographic change in species and even communities. That also allows them to correlate those patterns with natural and human caused changes in the environment. Unfortunately, though, sometimes certain taxa can be hard to identify to the species level or can even be misidentified, impacting how those specimens can be used. And those specimens and their accompanying data could be used for future research and conservation. And that brings us to my project, which is focused on these African tree frogs. African tree frogs are part of the family Arthroleptidae and belong to the genus Leptopelis. They're a group of charismatic big-eyed tree frogs that are endemic to sub-Saharan Africa, as we can see on this map, where all the green is. There are 54 known species, but there are still studies going on related to the basic taxonomy and systematics within the genus, including describing species that are new to science. So what's the problem? While Leptopelis have morphological differences that can help tell members of the genus apart, when in the field, species can look very similar to each other, which gives rise to them being misidentified or unidentified in natural history collections. In particular, Leptopelis exhibit extensive intraspecific variation in morphology, including coloration and pattern, as we can see depicted here with the Leptopelis calcaratus. They all are the same and from the same area, but they all look really different. Other features that can vary are the presence and absence of glands, the spurs, which calcaratus does have, little spurs at the end of the heel. Oh. Here. The Academy's collection has over 80 specimens that have tissue samples available for genetic analysis that are only identified to the genus level Leptopelis spa, and spa meaning a Leptopelis species. So what was my goal? My overall goal was to provide updated IDs for the Leptopelis spa in the collection, and my objectives were to sequence and align the 16S mitochondrial gene, which is a commonly used barcoding gene for African amphibians, for my Leptopelis spa specimens, then blasting my samples, which means to compare them with an online database of genes. And in cases where there wasn't a close match, I compared them to some unpublished sequences from Rayna and her collaborators, and then seeing what reference sequences matched closest to my samples. I then wanted to compare my specimen morphologies with their closest species match morphologies using field guides and examining specimens in the Academy's collection. And then I wanted to compare my updated species IDs to known distribution of species based on field guides as a final check for my species identifications. So how did I do it? First, I extracted DNA and sequenced the 16S mitochondrial gene for all of my mystery frogs. I then compared my cleaned, edited sequences, which I cleaned using Genius, to the public genetic database NCBI GenBank to see what published sequences were a close match to my mystery frogs. You can see here, you cannot see here. As you can see here, this specimen matched 100% to a Leptopelis blangeray, which is this cute little frog right here. But that's the first step. So we'll see what happens. Genius was also used to create a neighbor joining phylogeny to compare reference sequences for all of my species, for all species of Leptopelis that have a 16S sequence in GenBank, plus the ones from Rayna and her collaborators that haven't been published yet, and all the samples that I sequenced this summer. And I edited it using Figtree. So once I had the closest species match using my 16S mitochondrial DNA barcodes for my specimens, I further analyzed them by looking at each specimen's morphology and compare that to the known species morphologies for their match.
Some of the diagnostic morphological characteristics that I looked at to help confirm my specimen identities were the white spot under the eye, which is depicted by the white arrow, the tubercle under the tympanum, the presence absence of the tubercle under the tympanum, which is where it would be with the orange arrow, and then the width of the tympanum compared to the width of the eye. On the top specimen here has all of those things. It has a white spot under the eye. It has a tubercle underneath the tympanum being the eardrum of the frog, and the width of the tympanum is more than 1/2 the width of the eye. My specimen down here has the white eye spot, does not have the tubercle underneath the tympanum, and the width of the tympanum is about less than 1/2 the width of the eye. So all of these three characteristics found here this is diagnostic for Leptopilus boulangerii. So this shows how having a combination of these different morphological characteristics is important when confirming the identity of the specimen to the species level, because even when these possibly being defining characteristics, there are times when descriptions and field guides will say, has a white spot under the eye, but sometimes doesn't. So it makes it really hard to focus on just one characteristic. And there's still ongoing research to try to find better defining morphological traits that can help solve this confusion. So my final step was to then look at known species distributions, which is where the species is found geographically for the species IDs of my specimens, and compare that to where my specimens were collected. In this case, the specimen was collected. Okay, guess I don't know. Oh no. Okay, so in this case, my specimens are collected from Cameroon, and we know from the genetic and morphological data that we have a Leptopilus boulangerii. And to further confirm that, we looked at the field guide, and both are found in Cameroon. Leptopilus boulangerii is found in Cameroon, so we can be sure that this is indeed a Leptopilus boulangerii. Looking at species distributions can also just help identify if there is an incorrect species ID or indicate that the species occurs in more places than were previously known. So my results, which you got a sneak peek of. This is a neighbor joining phylogenetic tree of my 16S mitochondrial sequences for Leptopilus bus specimens with GenBank and unpublished reference sequences from Rayna and her collaborators. The colors correspond to the closest reference sequence that they match to, and with the combination of these genetic, morphological, and species distribution data, I was able to identify most of my specimens to the species level. Overall, I found 12 species from three frog families that are represented in my samples that I worked with. And now I'm just going to go over some of the few examples of some of the few types of results that I found. So 3 of my DNA sequences matched that I blasted through GenBank did not reveal the identity of Leptopilus spa, but instead showed that the closest match was to the species in the genus Hyperoleus. As you can see right here from my tree, they did not match to any. The Leptopilus. Hyperoleus are part of the family Hyperolidae, which is right here, and they are sister to the family Arthroleptidae. And they're also found in the same place. They're both endemic to sub-Saharan Africa, and Hyperoleus are also tree frogs. So they are found in the same area. They are tree frogs, but they are typically smaller than Leptopilus. So adult Hyperoleus could sometimes be confused for juvenile Leptopilus for researchers who are not very familiar with them. And in this case, after the sequence matched Hyperoleus and after analyzing the specimen's morphology, it was pretty clear that due to the elongated snout and the arrow-shaped body, and this is Leptopilus that does not have those characteristics, that this was indeed a Hyperoleus and not a Leptopilus specimen. And for some frog species all around the world, not just within the genus Leptopilus, we still don't know what their tadpoles look like. This is where DNA barcoding could be a really useful tool for matching larval and adults of the same species. I was able to obtain sequences for 2 Leptopilus bud tadpoles. One of them matched to a Leptopilus motawate, which is right here. You can see one of my tadpoles. That's a tadpole. Dawate is listed as an endangered on the IUCN list due to deforestation. And in order to protect habitats that a species relies on, we need to know what habitats they're using for each life stage. And in general, it's helpful to know what tadpoles of a species look like and know where they occur, because it helps us understand the reproductive biology of the species and identify what types of habitats they need over the course of their lifetime. And here we have what tadpole looks like. This is our adults. Leptopelis stolate. And my second tadpole did not match to any DNA barcode reference sequence on GenBank. And looking at the phylogenetic tree, their closest match was to Leptopelis bavomacatus and Leptopelis mozambicus. So our specimen was collected in Cameroon, so we can be pretty sure that it's probably not one of them, because this specimen is found from West Central Africa, and then we have South Africa, more of Eastern Africa. So what happened? Why didn't it match any reference sequence? A few reasons could be that this is a potential new to science species that we don't know about yet, or a species that just doesn't have a reference DNA barcode yet. So why is this information important? First, we were able to update the IDs of about 40 specimens to the species level with high confidence and corrected some pretty major mis-IDs in the collection. So this means the specimens and their data could be used more effectively for conservation and research purposes. The main limitation I came across in this project was having reliable reference genetic sequences to compare my samples to. And this is really important part of DNA barcoding, and is a challenge in parts of the world where the diversity of species is still an active area of study. So many of my GenBank sequences of Leptopelis have not been updated with the current species names, and many species have yet to be sequenced. So hopefully that will improve in the future. And my research highlights how much we still have to learn about Leptopelis and other African frogs, which is exciting because then I can do research on them. DNA barcoding could also potentially help us identify more reliable characters for telling these species apart in the future. And these data also serve as an important baseline for monitoring how species are adapted to global change and enable future research on these poorly known charismatic frogs. And then looking into the future, a couple of my genetic samples didn't work the first time, so I went back into the lab. Now I'm awaiting the return. Stay tuned. I plan on helping Lauren update the herpetology collection records and labels in the collection catalog, and maybe update some GenBank records that were problematic. And I plan on hopefully continuing frog research and helping to improve the herpetology collections at Humboldt when I return to school. For my acknowledgements, I want to thank the National Science Foundation for funding this program, California Academy of Sciences for hosting us, Rayna for being such a great mentor and guiding me throughout this whole process, Lauren and Rebecca for making this program possible and directing us, Athena, Lynn, and Grace for showing me everything I had to know about lab work, Lauren Scheinberg for allowing me access to the collection and also letting me ask all of the questions, Ed for supervising this program and being there for when I needed my sequences or any other question that I had, my fellow SSI interns for the support, and the new friendships. I'm really going to miss everyone. And the curators and collection managers for the interesting talks and leading us into their world, Ayanna Brown for the grad school advice and scholarship advice, the African tree frogs for their sacrifice science, the AmphibiWeb for all the resources that I use, Alan Channing and Mark Odell for the field guide. That was a crucial part of my project. And then Claude for emotional support throughout this time. Thank you. Any questions?
Thank you, Arlene. That was wonderful. So what was your favorite part about your research project overall?
I think my favorite part was learning how complicated frog research could really be, and that's really exciting to be a part of that and learn, like, okay, I could help try to solve part of the mystery, and just learning more about frogs is my favorite part.
I have a question. So like mysteries aside, what do you think that the implications of this research are for like more applied aspects of biology or conservation? Like how do you think this information could be used, in other words?
Good question. Knowing what species are out there in a certain, like say Cameroon, in a certain area in Cameroon could help us know, okay, is this a biodiversity hotspot? How many different species of frogs? And then it's just Leptopelis, or are there 5 Leptopelis species, or just one? That really helps us know what areas that we can protect and where there's more biodiversity.
questions?
I have a question about the tissues you used. So you were selecting ones that actually had tissues in the collection, correct? Were those tissues like preserved well for DNA extractions? And you mentioned there were a few that were tough. Were those like older specimens, like museum like collections, or how? Like, can you talk more about your tissue? Because I know museum collection tissues can be problematic for some of this work.
Okay, that's a great question. The tissue samples that I used they did have a really high DNA content in them, so I wasn't sure why they didn't work, but I am not sure if they were older or not. That is a great question that I can get back to you about. Thank you.
Great job, Arlene. Next up, we have Ethan Nee, who is joining us from the University of Massachusetts Amherst. And Ethan worked with Dr. Gary Williams this summer. Come on up.
All right. Good morning. Can you hear me okay? I'm not too close, not too far? Sweet. All right. Good morning, everyone. Thank you for being here today. My name is Ethan Nee. I'm a rising senior biology major at UMass Amherst, currently under the mentorship of Dr. Gary Williams here at the California Academy. My presentation today is entitled Morphology and Molecular Phylogenetics of the Rock Pens. So just hearing the title alone, you might be wondering to yourself what exactly a rock pen is. I'm pretty sure this is the right button. Okay, good. But before we answer that question, let's take a step back and answer the question, what is a sea pen? So sea pens are a highly specialized group of marine octocorals. And yes, they are animals. A lot of people look at them and think that they're plants. They're not plants, they're animals, like all corals. And taxonomically, they're in the order Pentathalacea, which is in the subclass Octocoralia, which includes things like sea fans and soft corals, and our sea pens, which is in the class Anthozoa, which contains our octocorals as well as our hard stony corals, and anemones, which is inside the phylum Cnidaria, which includes everything we just mentioned, plus jellyfish and other animals, which is inside the kingdom Animalia, which includes every animal, like this snake, and me, and you, hopefully. Any plants out there today? Anyway. So what's the takeaway from all that jargon? Basically, all of that boils down to these sea pens being colonial animals that are comprised of many, many polyps living together. There's actually two ways you can refer to a polyp. You can either think of it as a single organism with many mouths, or as a colony of individuals where the individuals are the polyps. So yeah. Oh, I also want to take this slide to sort of highlight some important sea pen anatomy that you're going to need to know for the rest of this talk. So if we look at this species in the middle, this is Tylocercus gurnii. Probably, if you're familiar with sea pens at all, this might be one that looks familiar. This is probably one of the more famous ones. But you'll notice that it's split into two parts. The top part is called the rachis, and the bottom part is called the peduncle. The rachis, the top part, is where most of the polyps live. So this whole portion is made of individual polyps, and this is the part that's actually exposed to the water so that they can feed. The bottom part is the peduncle, which is sort of the way the sea pen is attached to its substrate. In most cases, it's soft sediment, so it has this long peduncle to help it get buried so it doesn't get washed away in the currents. Okay, so that was a lot. Let's take a second. I'm going to give you guys a break to admire some cool sea pen gifts while I take a drink of water. Okay, I just think they're cute. Anyway. Now that we're aware of what a sea pen is, we can go ahead and answer the question, what is a rock pen? So rock pens are sea pens that have evolved an adaptation that allows them to bind to rocky surfaces. And before they were discovered, it was believed that all sea pens only inhabited soft sediments like sand or mud. But it was discovered or documented in 2011 in a paper in the journal Zootaxa by Gary Williams and Philip Alderslade, that there are 4 species of sea pens that exist that can bind to rocky surfaces or rocky substrates. 3 of them are included in this study. This is a diagram from that paper. You'll notice that there's 2 peduncle structures for 2 different species. One of this If you notice the one on the left, this is a more traditional C-pen peduncle, and this one on the right is a rock pen peduncle. You'll notice that the difference is that this one is a lot longer, it's a lot more narrow, and it's sort of like sausage shaped. And what that does is that it allows it to bind well to sandy substrates and burrow itself and anchor itself into like mud and sand. If you notice the rock pen peduncle, there's also a picture of it here. You'll notice that it's a lot wider, it's a bit shorter, and the reason for that is because these peduncles essentially act like suction cups or plungers that allow them to bind, sort of like make a seal on those hard surfaces. Okay, so the goal of my research was ultimately to uncover how rock pens are evolutionarily related to other sea pens and octocorals, with the ultimate goal to be to visualize the evolutionary relationships between these species and other octocorals through the construction of a phylogenetic tree or an evolutionary tree. And like I mentioned in the last slide, we looked at 3 species of rock pens, Anthoptilum gallet-holmesii, an Anthoptilum species from Alaska, and Calibolemnon francii. Okay, here's a complete list of all of the species that we looked at. And the reason, including the 3 that we just mentioned, which are highlighted in orange. And the reason we looked at these species in particular, there's 10 of them, is because we wanted to sort of cast a sort of wide diversity of species within Octocoralia, so we can really nail where they are in the evolutionary tree. This list of species includes those that we hypothesize to be very closely related to the rock pens. Okay, so my research used 2 main sources for phylogenetic information. The first being genomic analysis, which was done in the Academy's Center for Comparative Genomics, or CCG. And then the second source being morphological analysis of the sclerites, which was done in the Scanning Electron Microscopy Lab, or SEM. And all of the samples that we used came from the Academy's collection of invertebrate zoology. And this is an actual picture of all the specimens that we used. So you get a good idea of sort of the diversity in size and shape and color and just general form within all the octocorals. So getting into morphology first. So these are sclerites. These are images that I took using the SEM of soft coral sclerites, or octocoral sclerites, excuse me. So essentially what sclerites are these tiny little dust-sized particles that are made of calcium carbonate found in most octocoral species. And the reason they have them is because in contrast to hard stony corals, like the reef-building corals that you might be more familiar with, the corals that we look at don't have those. So they rely on these tiny little dust-sized particles within their tissue to help maintain their structure. And they can be helpful for morphology. So these are 3 species that I was able to image: Chromoplexura marcae, Hersemia julipachardae, and then species of Umbellula. So just looking at these images, what kind of phylogenetic information can we derive from these sclerites? Well, as it turns out, not much. So yeah, anyway, it turns out that sclerites are perhaps best used for species identification and description, and sclerites alone cannot provide enough phylogenetic information, at least for the analysis that we're looking for. And it was previously observed that in some sea pens, including the rock pens that we're looking at, have very reduced sclerites, which means that they're sort of these minute ovals, and there's very few of them. So they're sort of tedious to work with, and they're not very rich in information, so they're sort of not worth the trouble of going through analysis. So then, where can we turn for our phylogenetic information? This is where molecular genomics comes in. So my research focused on the sequencing of 3 genes for each species, the genes being ND2 and ND6, which are mitochondrial genes, and MSH1, which is a nuclear gene. And the reason we looked at multiple genes rather than just one for each species is because if we looked at 3 genes and we analyze and sequence those 3 genes, it gets us more data back so that we can construct a more robust analysis. And if you look here on the right, this is a schematic of sort of from top to bottom, it's a schematic of the workflow that we did in the CCG. So we started with DNA extraction to isolate the DNA from the tissue samples that we got from the preserved specimens. We did PCR amplification to increase the amount of DNA to multiply and amplify it so it's sufficient for analysis. We then used gel electrophoresis, which is shown down here, to check the PCR product to make sure that the reaction worked properly. And if it did, we moved on to purification using exosap, and then we outsourced for Sanger sequencing. And essentially what they do is they take your DNA, They run it through a machine and they give you a file back that shows you the sequence of the base pairs, like your A's, your C's, your T's, your G's. So we took that file, or those files, there's a lot of files. We aligned them and edited them using a software called Genius. And then we made a maximum likelihood phylogeny using IQ-Tree, and then an actual figure using a software called Figtree. Okay, I'm gonna get more water while you look at more pen videos. Okay. Moving on. Now that I have your attention, we can move on to results and discussion. So first, this is our phylogeny. It's a maximum likelihood phylogeny that we made using IQ-Tree and Figtree, as I just mentioned. In red are our 3 rock pen species. And then you'll notice that in blue, this is the Anthoptilum genus, which contains all of our rock pen species. And if you look here, it also includes a non-rock pen species, which is important. Okay, so going off of this phylogeny, we can conclude that all the rock pens that we studied form a monophyletic group within the genus Anthoptilum. We can see that they diverge from a common ancestor to form a sister group with the non-rock pen species within the genus Anthoptilum, which is represented here by Anthoptilum grandiflorum. And lastly, we also found that Calibelemnon francii, which is highlighted here in orange or in yellow, excuse me, is actually a species of the genus Anthoptilum, like all other rock pens, and should be considered for reclassification based on this tree. Okay, so future research will sort of dive into more of the genomic work to construct a more robust analysis, including adding more rock pen species to our phylogeny, namely Anthoptilum lithophyllum, which was supposed to be included in my analysis, but was cut because we were having trouble doing the PCR and the extraction in the CCG. So if we had more time, we would have made it happen, but we were working on a tight deadline. Additionally, future work could be done to better understand the relationships, the evolutionary relationship between the genus or the rock pens and the non-rock inhabiting species of the genus Anthoptilum, including finding divergence time between the common ancestor of the rock pens and the other Anthoptilum species. Okay. Thank you to the National Science Foundation for funding this program. Thank you, Dr. Gary Williams, for your unwavering support and guidance throughout this project. Thank you, Dr. Lauren Esposito, Dr. Rebecca Johnson, Dr. Ed Myers for your support in making this happen. It's been a really fantastic experience, and I don't think I could ask for more from a program like this, so thank you. Thank you, Dr. Athena Lamb, Lynn Bonomo, and Grace Kim for making the CCG run beautifully smoothly and being really patient when I inevitably asked a lot of questions or messed something up. Thank you, Dr. Bruce Byers and Dr. Felipe Salas for helping me in my application, for being references. And thank you to my fellow SSI interns and the rest of the Cal Academy staff that I've interacted with while I'm here. It's been really inspiring getting to work with all of you, and I'm excited to see where all of us go in the future. Thank you. That was okay on time.
I'm curious if you have any guesses about why that one species or sample wouldn't amplify.
Yeah, we probably had to do more troubleshooting to figure out why. It could be just the sample that I got just wasn't a good tissue sample to use. It could also be the age or the ethanol that it's preserved in was not great. Yeah, so probably more troubleshooting to be done to figure that out.
I was wondering, for the rock pens,
Yep.
do you know why they have reduced skeletal elements? Like, is there something different about the way that they're moving or the habitat where it doesn't matter as much to be so sturdy?
Yeah, that's a good question. I don't know, like concretely. I can't like concretely say. Dr. Williams, feel free to chime in if I'm saying anything that's incorrect. But I don't know. It could be just something with the structure and the pressures that they're subject to. Just doesn't require them to have sclerites. Perhaps at the depth. I'm not entirely sure. It's the sclerites and the reason that they're reduced in those rock pen species. Yeah, okay. Yeah, so Right. Yeah. So if I go back all the way to the beginning, or actually, if I go back, if I look at this one, this will show sort of. So C-pens, a characteristic of C-pens is that they have this sort of internal axis that sort of differentiates them from like soft corals and stuff like that. And the reason that they have that internal axis is for structure. So I guess if they have that, then some of C-pens have evolved to just rely on this rather than the sclerites for structure.
You just briefly mentioned something about depth and pressure, and I was wondering what depths your samples were collected at.
Yeah, it's actually, I don't know like the concrete numbers per se, but I know that there's at least a decent diversity in terms of depth. I know I think Anthoptilum gallet holmesii, I think the one that we got was found around, or it can be found around 2,000 meters, so relatively deep.
Awesome, Ethan. I didn't know about these rock pins. I think it's amazing to learn about them. I'm wondering, you talked about the structure that holds onto the rock being kind of like a plunger or a suction cup. Is it actually using suction, or does it have some other way of adhering to the rock?
Yeah, so I believe that the way it works is that they're, again, I could be wrong since anatomy wasn't my focus, but I believe that they attach to the rock and they sort of, I think, suck up some of the water to create a seal that acts essentially like a plunger. Is that, I can confirm that. Yeah, yeah. So yeah, it's like a seal that it makes. Yes, Dylan.
So do they have any predators?
Yeah. Oh, yeah. Okay. I'm really glad that I put this in now. Backtrack. Almost there. There we go. Yeah. Okay. So, I guess there's, as far as I'm aware, there's two main predators for that prey on sea pens. The first being like nudibranchs, and the other being sea stars. So, there's a nudibranch feasting on a poor little sea pen there, and the reason it's shrinking there is because it helps it like bury itself back into the sediment to escape predation. Hope that answers your question.
I had another question about the anatomy. Sorry.
Sure. Yeah.
So the central structure and how it's attached, is that like a living part of a polyp? Is that something that they produce? Like, yeah, just curious about.
Yeah. So, I guess throughout the life cycle of the sea pens, the initial polyp is essentially what becomes the peduncle, like the first polyp that all of these grow from. All of these tiny secondary polyps come from, is essentially what was at one point the initial polyp, like the first polyp to like land, and became the peduncle. And I guess my assumption is that it extends up into becoming the internal axis, although I cannot definitively say once again. But yeah.
Thanks.
Oh, does Sophia have a question?
Sorry.
Or I don't know if we're over time, but.
Actually, Parker had a question back here, and then I'll bring it up.
What's a good question today?
So the one rock pen species that was formerly classified as a different genus,
yep.
Was that previously considered to be its own genus, or is it was it previously considered to be related to some other group of sea pens? And do you know what the reason is why it was classified incorrectly before?
I'm not entirely sure. Sort of answers to the both of those questions. I don't know if how many other species are in that genus, if there are any others. But if I had to guess, I would say that I don't think that there was genetic or genomic work that was done on that species before. So that could have had something to do with it. It could have been morphology that sort of led them in one direction, whoever, described it. That would be my guess. Yeah.
My question is, what was your experience like working with SEM? And in the future, do you see yourself doing more morphological or genetic work?
Yeah, so it just so happens that the sea pens that we were looking at don't provide much information in terms of the morphology that we look at under the SEM. There are other octocoral species, like the ones that I took pictures of, which is, nope, almost there. There we go. Yeah, like these ones. These ones can provide information just because they're not just little ovals. So yeah, if we're looking at these species and then we see the sclerites, we can make inferences, I guess, based on just the morphology. It just so happens that the sea pens that we're looking at don't have sclerites that are particularly useful. Thank you. Stick around for Nelly's talk.
right. Thanks so much, Ethan. Oh, I have to go forward more. Hold on. It's okay. All right. So next up, our last talk for the morning, we have Nelly Batista, who joins us from American River College. And this summer, Nelly's advisor was Dr. Jennifer Hoey. Come on.
Okay, hello, good morning, everyone. My name is Nelly Batista. This summer, I worked along with Dr. Jennifer Hui with my project as a non-invasive micromorphological approach using image analysis for the diversity identification of madrasas in Curacao. Okay, so I'm going to begin my presentation talking a little bit about corals and what are some of the morphological characteristics that define specifically scleractinian corals, which I am working on within my project. So scleractinian corals are made up of calcium carbonate and contain a thin layer of tissue. So as we can see in this image, half of it shows the hard structure of the coral that is made up of calcium carbonate, and the other one is a thin layer of tissue where the polyps and the tentacles are present, and the rest of the coral's anatomy structure. Polyps are actually small marine animals. Corals are made up of many polyps that make up what we know as a colony, as we can see here. Every polyp sits in a calcium carbonate structure called a corallite. And something very important about corals as well is that they have a mutually beneficial symbiotic relationship with algae, also known as zooxanthellae. So algae provides corals with all of these beautiful colors that they come in and allows corals to live successfully in tropical waters that are otherwise very nutrient and food poor. And as a return, algae can live in the coral's tissue, allowing both organisms to survive better together than alone. So why are corals important? Corals are, one of the main things is, well, they are among one of the most important animals in the ocean for several reasons. First of all, biodiversity. Coral reefs are one of the most biodiverse ecosystems on the planet, supporting nearly 1/4 of the world's marine species, despite covering less than 1% of the Earth's surface. So they do a lot. Another thing is food and tourism. Healthy corals support commercial and sustained fisheries, as well as jobs and business through tourism and recreation. They are also great for coastal protection. So they buffer shorelines against 97% of the energy from waves, storms, and floods, helping to prevent loss of life, property, and erosion. And this was kind of new to me as well, but this biodiversity is considered a key to finding new medicines for the 21st century. Many drugs are now being developed for coral reef animals and plants as possible cures for cancer, arthritis, human bacteria infections, virus, and other disease. So essentially, they are tiny superheroes of the reef. So what is happening to corals? Many of us know corals as the rainforest of the sea. They are, as I mentioned before, one of the most diverse habitats on the planet, but unfortunately, they are bleaching. So there's many threats to this. They are facing a lot of pollution, disease, and habitat destruction, and elevated sea temperatures. So in other words, global warming. Corals are just like humans. They also become stressed, and this happens when water is too warm. So the corals will essentially expel the algae that lives in their tissue, causing the corals to turn completely white. When a coral bleaches, it's not dead, but the repeated bleaching can lead to corals starving to death due to no longer having the support of the algae to provide them with oxygen and nutrients. And unfortunately, once corals are damaged, they are less able to support the many creatures that inhabit them and the communities that live near them, and many of these reefs never recover. So this brings me to my topic. I'm going to be I worked with madrasas and its morpho species. So M. carambi, M. decactus, M. perensis, and M. cinerea. So an objective of the study is to quantify 2 morphological traits to further understand madrasa species in different environments. The way that we did this was through macrophotography. So essentially what a macrophoto is, it's a photo taken underwater showcasing a coral larger than it is in real life. In the past, a lot of these morphometric studies required a large collection of skeletal fragments. this was like super invasive, time consuming, and a risk to compromise the coral's fitness, since it would leave it exposed to a lot of virus and bacteria, causing disease in the damaged colony. And through microphotography, we can zoom in as much as we want and analyze all the traits that we would like. And most importantly, it is non-invasive, so we are avoiding any damage to our already threatened corals. So here is an image that kind of like represents how macrophotos are taken. This is Dennis, who is part of the Reefscape Genomics Lab. He's getting very close to a coral colony, which is the way that we collect these macrophotos, and so I can further analyze them. So the locations where these macrophotos were collected were in Curacao, which is a Dutch Caribbean island close to Venezuela. The data was collected from 7 different locations in Curacao at 5, 10, 20, 40, and 60 meter depth. My focus was on coral state, where there was a collection of 251 macrophotos, and over the summer, I successfully analyzed 104 of those. So apart from macrophotos, we also have something called photogrammetry, which is essentially a long reefscape. The Reefscape Genomics Lab were able to obtain a 3D model plot that was taken from a 25-meter long coral reef. So it's kind of like the map of the reef. The way that photogrammetry happens is that a large camera is basically taking a lot of pictures per minute, allowing us to put them together and obtain an overall of a 3D image that later becomes digitalized and can be zoomed in and analyzed to identify specific colonies. Here's an example of kind of how it happens. So essentially what this 3D model allows us to do is to track coral change over time and its specific locations, along with genomics and morphometric data, be able to return to the reef and observe how our coral colonies are doing over time and its reaction among changing environments. I do want to clarify that the image is just a reference, how we can use the locations of each colony, but if we zoom in, we would not be able to see, okay, we would not be able to see specifically like this image. It was just like as a reference, so if we know where each colony is located if we were to return to the reef. So the way that macrophoto analysis happens is, as we can see in the first image, I use these white little squares as a reference. So each square I would measure it, and it was 1 millimeter. And for the second photo, I would use this yellow ruler, which is essentially a 5 millimeter reference. And this is the way that I collected my data. So 2 of the traits that were analyzed were septa count and intercorallite distance. And as we can see from the first photo, there is an outline of corallites in each madrasis where septa surrounds like the corallite area. So basically all of these little flowers are corallites and septas is everything that surrounds them. So data collection. So the data was collected through an image analysis program called ImageJ. Generally, depending on the coral's morphoid species, its septas per corallite could range from 6 to 10. I focused specifically on septa counts. So as I mentioned, I would choose 15 corallites per image, and I would count its septas and analyze them. So basically the little numbers that on top are just like what I would use to number them and reference like the corallites that I was going to go back and further analyze. And in the second photo, we have intercorallite distance, which is basically, I would choose 5 groups of 3 corallites and measure the distance of intercorallite distance between adjacent corallites as well. Okay, so after collecting my data, I was able to export it into R and code my results through a series of box and whiskers plots as shown here. I'm going to show you a series of my results through plots for intercorallite distance of madrasis morphospecies through boxes. So as we can see, on my y-axis, I have an average of intercorallite distance, and on my x-axis, it's the depths that it was most commonly found. The N just represents like the colonies and the morphospecies that I analyze per depth. So I did that for Madrasis carambi, Madrasis decactus, as well for Madrasis forensis, Madrasis cinerea. We can see as well where the depths were mostly located. And we also had an unknown plot. So these are species that are shown to be similar to other morphospecies, but along with some incoming data, we'll be able to categorize them. In the last plot, we can observe all the species together to compare its intercorallite distance among its designated depth locations. So we did observe a pattern. So at deeper depths, intercorrelate light distance were shown to be greater. So as you can see here, the deeper it gets, the greater the intercorrelate light distances. I did this as well for the average septa count for each Madras's morphospecies. And as well, I used average septa count as my y-axis and depth for my x-axis. And I did that for Madras's Carambi, Madras's Cactus, and as I mentioned previously, the n just represents the amount of colonies that I analyzed per depth. I did the same for Madras's Francis, Madras's Cinerea, and I also had an unknown box as well. So based on the overall septa count for Madras's, we can observe that Madras's Cinerea has the lower septa count compared to other morphospecies. So technically, it's like the most different one. So this brings us to our overall PCA results. So this PCA graph demonstrates a reduction of highly dimensional data sets by summarizing its variation through its 2D principal components. Each plot shown on this graph represents an analyzed individual colony categorized by colors depending on its initial field species identity. In this PCA, it includes an overall of 30 traits that were analyzed per colony with 50 septa count, plus 15 intercorrelate distance measurements. So what this PCA is doing is it's essentially capturing the closeness of each colony represented by a plot. The closer the points are from each other demonstrates the greater morphological similarities within species, and the further they are just demonstrates greater differentiation within species. So after some PCA analysis were done, we observed a general pattern of 4 clusters of morphological similarities. A preliminary pattern observed within these PCAs was that, as you can see, Madras's Cinerea, which are the orange dots, seem to have a greater morphological differentiation compared to other species, which is actually consistent to some genomic data as well. So in conclusion, now you might be wondering what septa count and intercorrelate distance has to do with helping the conservation of our reefs. So some preliminary patterns were able to be observed, but greater data collection and analysis is essential to support any hypothesis to further understand and help our reefs. During the summer, I worked on building this data set among madrasas as a start to understand its biology and how to differentiate each morphospecies and how we know and how we know a fundamental aspect of conservation is knowing what you have to find ways to protect it. Through this research, we are further understanding corals, specifically madrasas, by comparing morphological analysis and genomic data, with the goal of observing any hybridization and further understanding it. Once any hybridization is observed, we will be able to study its reaction among specific environments and track if its variation is beneficial or detrimental to our reefs. The scale of solutions needs to match the scale of the problem, which is why collective actions are required to help our reefs. Our reefs continue to undergo, unfortunately, massive bleaching events due to human impact, and our job as a community and scientists is to help these beautiful species to continue to live and support our wildlife for more healthy and colorful oceans. So looking into future directions, my involvement in this research project has awakened a new passion within me for the conservation of our reefs. Furthering my education within a narrowed area for core research is a plan as well. And I will be continuing my involvements within the Rescape Genomics Lab to further research corals as well. So I do want to give some acknowledgements. I would like to thank our program directors, Dr. Lauren Esposito, Dr. Rebecca Johnson, and our supervisor, Ed Mayers, for making this summer possible, along with your motivation to continue research. I would also like to thank my mentor for always being so encouraging and patient, as well as a great part of my professional development. I would like to thank the Rescape Genomics Lab for being so supportive and welcoming to me through this whole summer. Other curators and staff members that made all tours and workshops possible and interesting. And Livia Nelly for allowing me to use her thesis as a reference for my project, the NSF for funding this wonderful program and making all of this possible, and a special thank you to all of my SSI peers for making this summer a memorable experience. Thank you. Any questions?
Awesome job, Nelly. I have a quick question about the unknown that you looked at, or the unknowns, and I'm wondering if those might be more than one species. Like if it was one unknown, if it was multiple unknowns. I don't remember like how many individuals you looked at, and whether or not. So the question, like, are they different species potentially because of the way they grouped out? They were really variable. Like it was the most variable thing that you looked at, and also is it a potential hybrid? Like, what's the story with the unknowns?
Okay, so the unknown is basically it is within the madras's range, but when the divers went into Curacao, some of them were like identified, kind of like just like within like other madras's, like Francis, the cactus, or so. We're still waiting on some genomic data as well to kind of like back up any information that we have. We do have a specific like range within like septas that we're able to like categorize them and be like, oh, this might be, you know, within this category. But genomic data will be best to clarify all that information. And within hybridization, we are still in the process of that as well. We are along with some genomic data as well. We're going to be receiving and comparing that to my graphs as well. Some of my graphs were within the range. Some of them actually exceeded it. So some coralites had even up to like 16 septas, which could demonstrate some type of hybridization, but we still need more data to back that up. So yeah, we will continue the search, but that is a wonderful question. Thank you.
Nelly, that was an awesome talk. Thank you. As you continue with the Reefscape Genomics Lab, what are your next big questions, or what is the thing that you really want to explore next?
So I am going to continue doing some more image analysis. We are waiting on, as I mentioned, Jennifer's genomic data, and we are planning on comparing both of them and kind of like seeing if there is any hybridization present, and if not, then we still have some morphological analysis that will help us to kind of like categorize these madrasas and see if we can like fit them within a specific range that varies from what we already know. So yeah, we will continue the search, but so far, yes, I will continue doing image analysis, which is really cool because I look at really pretty corals all day. But thank you.
What are you most looking forward to in terms of next steps, like with the additional data that will come in?
I'm looking forward to, I do have like a couple of hypotheses that I have thrown around every here and then. But as I mentioned, just like any other science, we still need some more genetic information, some more data to be able to confirm any of this information. But I am really looking forward to be able to identify any hybridization and see how they react, because if we identify one, we go back and we see that maybe around this hybridized coral, a lot of other ones are bleached, then we can observe what it's doing and how it's reacting, like compared to other corals, and how we can further like help it. So maybe it adapts to warmer temperatures, to more acidic waters. So yeah, essentially, hybridization would be like the door to finding out more solutions to helping our reefs as well.
Thank you. So let's just give all of our morning speakers another round of applause. And we will be resuming at 2 o'clock. There's another link if you're joining online. There's another link in the meeting invite to join that second session. And we'll see you back in a couple hours. Thank you.